Okay, hi everyone. My name is Laurent Deluc. I'm associate professor in the Department of Horticulture at Oregon State University. And today I'm going to discuss our project that was funded last year, which is to evaluate the impact of double-strand RNA molecules mediated silencing on the 4 grapevine host factor and how it could potentially impact the activity of the grapevine red blotch viruses. I will go through a few slides to introduce the topics, show you actually some prelim data and how we build this project, the conceptual approach to validate our questions, the methodology and the current study of the experiments, and some perspectives. Okay, in terms of introductions, grapevine red viruses as a Gemini virus is no exception regarding the need of interacting with host factor to survive. So 3 years ago, we got actually awarded for a project which was basically to identify some interactors in grapevines that were associated with all the replication-associated proteins. So here is the grapevine genomes. And you can see that we were trying to identify through some genomic screens potential interactor with the replicase and the C1 and C2 and C1, C2 together and C3. So from that, the only data we gathered was actually the characterization with a high confidence about 4 host factors that were interacting with the replicate of GRBV. These are actually the name of those host factors. At that stage, they are just interactors. We don't know actually their role as either pro or antiviral factors is unclear, and this is why there was a need actually to develop a new project trying to address these questions. The characterizations of the host factor were done by genomic screen. The growth of the yeast here is just indicative of actually the interaction between the C1 and those host factors. And we also validated a few of them in plant data by a different test, which was basically the same idea. When you do have the interaction between the host factors and the replicase, you do have here a yellow signals that is detected in grapevine leaf cells. As you can see here for the chloroplast vesiculation proteins, and here with the BH-LH59s. Okay, so based on that, we do have this idea of the tend to be actually interacting. The idea would be to understand whether they are associated with the defense mechanisms or they are used by the pathogen to basically facilitate its multiplications. So either as a pro or antiviral factors. So one way to do it is basically to silence those genes and see how the GRBV is behaving. So the approach is simple, is actually to deliver double-strand molecules and that are specific to those host factors and that will trigger RNAi immune reactions in the plants, which will basically lead to their degradation of those degradations or the silencing of those genes. And then if the GRBV is actually in those plants, you can see whether or not there is a change in the viral load. So we design and basically devised the 2 objectives of the project that we got actually the awards. When in the objective 1, we wanted to measure the titer of the GRBV in plants that were infected with the virus, but they were also silenced for the 4 host factors. And the objective 2, if we do have actually a good sense of silencing is leading to a decrease in the GRBV load, we wanted to make sure that the silencing through the delivery of the double-strand RNA would not have actually an unintended effect on something else in the plants and ensure actually the plant fitness. And this would be actually using multi-omics approach. So that is the 2 objectives of the project. The methodology is basically something that we streamlined in my lab, which is basically using a pretty conventional way of delivering Gemini virus now, which is an artificial inoculation of the GRBV genome. So you can see here, this is something that was developed about 5 years ago now. And the idea would be to recover the plants and deliver on those tissue culture plants a population of double-strand RNA that basically are supposed to target the 4 genes associated with the interactors. And you are delivering by root sucking. And then it's just a matter of collecting the samples over a period of a month, and then do the different downstream analysis, like titering the virus, and potentially do the small and RNA sequencing to check the unintended effect on the biology of the plants. At that stage, in terms of where we are at in the project, we have basically infected all the plants with artificial infections with the viruses. 3 sets of 10 plants each were delivered with the double-stranded RNA targeting the BHLH and the chloroplast vesiculation proteins. And we do have also, as a control or as a mark, a series of plants that were delivered with an unrelated double-stranded RNA. Post-delivery sample collection has been complete for the mark and the BHLH. The one for chloroplast vesiculation is underway, and the delivery of the double-stranded RNA for PB1CP and fasciclin-like 2 proteins is supposed to be happening within the next month. From there, we will do the viral titer, and if the viral titer is showing actually a decrease in the GRBV expressions, or the amount of molecules of GRBV, then we will go with those candidates, plants or candidate experiments, and we will extract the coding and non-coding RNA, and we will run the RNA-seq data. That's pretty much it. We hope that the RNA-seq will be conducted by the beginning of June. Once we have clarity about the pro and antiviral roles of those host factors, the next step would be basically to test this into all plants by follow applications. I think most of you now should be actually pretty familiar with what we call spray-induced gene silencing. So the next step, let us say, as a matter of fact, in our lab, we have been awarded for over a project on the GRBV and developing foliar applications using a nanocarrier that enhance the systemic delivery of the RNA molecule and also improve the extent of silencing over a period of 3 months. Since last year, we have identified new series of interactors that could be tested in the future for their pro or antiviral properties against the red blotch. That's pretty much it. Here you have all the different literature cited in these presentations. And if you do have any questions, please feel free to contact me at my email address, delucl@OregonStateUniversity. Thank you very much.